Hello students, and welcome. I'm glad you could join me for this comprehensive session on embryology. Today, we're going to walk through the fascinating journey of early human development, from the very first cells to the formation of complex structures. This entire lecture is based on the detailed notes provided, so you'll be getting a complete and structured overview. I encourage you to grab a notebook and a pen, as following along and taking your own notes will be incredibly helpful. Let's begin. Our story starts with the primordial germ cells, often abbreviated as PGCs. These are the very precursors to gametes, that is, the sperm in males and the oocytes, or eggs, in females. These crucial cells originate from a layer called the epiblast, and they first appear during the second week of development. Initially, you'll find them located near the caudal, or tail end, of a structure called the primitive streak. From there, they follow a specific path, moving through the primitive streak to the wall of the yolk sac. Their migration begins in the fourth week, and by the fifth week, these primordial germ cells arrive at their final destination, the developing gonads, which will become the testes or ovaries. Next, let's discuss the concept of an organizer. Think of an organizer as a morphogenetic stimulus, a region of cells that induces, or signals, other cells to form new tissues. There's a hierarchy. The primary organizer is the blastopore, which induces the formation of the notochord. The notochord then acts as a secondary organizer, inducing the formation of the neural tube. Finally, the neural tube acts as a tertiary organizer, signaling the segmentation of somites. This leads us to a fundamental concept, stem cell potency. Potency refers to a stem cell's ability to differentiate into other cell types. We can rank this from highest to lowest potential. The highest level is totipotent. These cells can form all embryonic and extraembryonic tissues, like the placenta. The zygote and the early blastomeres are totipotent. One step down is pluripotent. These cells can form all tissues of the embryo itself, but not the extraembryonic structures. The inner cell mass of the blastocyst is a perfect example. Next is multipotent, which can form multiple, but related, cell lineages. Hematopoietic stem cells, which form various blood cells, are multipotent. Below that, oligopotent cells can form just a few cell types, like vascular stem cells. Finally, unipotent cells can only form one specific cell type, such as a liver cell. Now, let's turn to meiosis, the special type of cell division that creates our gametes. Meiosis involves two rounds of division. Meiosis 1 is called the reductional division because it reduces the chromosome number by half. It begins with prophase I, which has 5 substages, leptotene, zygotene, pachytene, diplotene, and diakinesis. This is followed by metaphase I, anaphase I, and telophase I. An important event called independent assortment occurs during meiosis 1. This is the random distribution and separation of maternal and paternal chromosomes, which is a major source of genetic variation in the resulting gametes. After meiosis 1, the cells proceed to meiosis 2, which includes prophase 2, metaphase 2, anaphase 2, and telophase 2. The final result of the entire process is 4 haploid cells, meaning each cell has only a single set of chromosomes. Let's focus on the female reproductive system and the ovarian cycle. The ovary contains several key cell types. Granulosa cells have FSH receptors and produce estrogen and inhibin. Theca interna cells have LH receptors and produce androgens, which are then converted to estrogen. After ovulation, the corpus luteum becomes the main source of progesterone and estrogen. Now for some key facts about oogenesis, the process of forming mature oocytes. This process actually begins before birth. The maximum number of oogonia, about 7 million, is reached at the 5th month of intrauterine life. At birth, this number drops to about 2 million primordial follicles. By puberty, fewer than 400,000 remain, and of these, less than 500 will ever be ovulated. Every month, about 15 to 20 follicles are recruited, but typically only one matures fully to form an ovum. In the male system, we have spermatogenesis. This process is supported by two main cell types in the testes. Leydig cells, found in the interstitial tissue, are stimulated by LH to produce testosterone. Sertoli cells are stimulated by FSH and play a crucial role in supporting the developing sperm cells. They secrete hormones like inhibin B and produce androgen-binding protein, or ABP. The germ cells themselves are dedicated solely to spermatogenesis. Let's look at the stages of meiosis in males. The primordial germ cells remain dormant in the testis until puberty. After puberty, a spermatogonium divides to become a primary spermatocyte. This cell undergoes meiosis I to form two secondary spermatocytes, which are haploid. These then undergo meiosis II to form four round spermatids. The entire process from spermatogonium to spermatid takes about 74 days. The round spermatids must then undergo a transformation called spermiogenesis. This is not a cell division, but a maturation process. It involves condensing the nucleus, forming a tail for motility, creating an acrosome from the Golgi apparatus to penetrate the egg, and shedding excess cytoplasm. The sperm finally attain full motility in the epididymis, a long, coiled tube attached to the back of the testis. Now let's revisit oogenesis. Before birth, oogonia develop into primary oocytes, which then become arrested in prophase I of meiosis. They remain in this state throughout childhood. From puberty onwards, a surge in LH causes a primary oocyte to complete meiosis I, producing a large secondary oocyte and a small first polar body, which typically degenerates. The secondary oocyte then begins meiosis II but arrests again, this time in metaphase II. This is the cell that is ovulated. Meiosis II will only complete if the oocyte is fertilized by a sperm. Following fertilization, we have a series of critical events. Let's trace the process from coitus to the acrosomal reaction. First, semen is deposited in the vagina. The volume is typically 2 to 5 milliliters, and its slightly alkaline pH helps with sperm motility. Immediately after deposition, the semen coagulates, due to an enzyme from the seminal vesicles. Within 20 to 30 minutes, it liquefies again, thanks to fibrinolysin from the prostate, which allows the sperm to become motile. The sperm then begin their migration. Aided by thin, watery cervical mucus present mid-cycle, they travel from the vagina, through the cervical canal and uterine cavity, into the fallopian tube. Fertilization typically occurs in the ampulla, the widest part of the fallopian tube. Along this journey, sperm undergo capacitation. This is their final maturation step, where the glycoprotein coat is removed from the acrosome. This process takes 6 to 8 hours and results in hyperactivation, making the sperm more motile and its membrane more permeable to calcium, preparing it for the next step. Sperm can survive in the female reproductive tract for up to 5 to 7 days, but they are most fertile within the first 24 to 48 hours. To reach the oocyte, they must first penetrate the outer layer of cumulus cells, called the corona radiata, using an enzyme called hyaluronidase. Then, they must bind to the zona pellucida, a glycoprotein layer surrounding the oocyte. This binding occurs at a specific receptor called ZP3. Binding to the ZP3 receptor triggers the acrosomal reaction. This involves the fusion of the sperm's plasma membrane with its outer acrosomal membrane, releasing powerful hydrolytic enzymes like acrosin. These enzymes digest a path through the zona pellucida, allowing the sperm to reach the oocyte membrane. Once one sperm fuses with the oocyte undergoes the zona reaction, altering the zona pellucida to prevent any other sperm from entering. Once fertilization is complete, we have a zygote. This marks the beginning of pre-embryonic development, which covers the first 2 weeks. Within 24 hours, the zygote begins to divide in a process called cleavage. By day 3, we have a compact ball of 16 cells called a morula. Around day 4 or 5, fluid enters the morula, forming a cavity called the blastocele. This new structure is the blastocyst. The blastocyst has two distinct cell populations. An outer cell mass, the trophoblast, which will form the placenta, and an inner cell mass, the embryoblast, which will form the embryo proper. Around day 6 or 7, the blastocyst hatches from the zona pellucida. By day 9, it has implanted into the uterine wall, and by day 12, the bilaminar disc has formed. Let's look closer at the second week. On day 8, both the trophoblast and embryoblast differentiate. The trophoblast splits into an inner cytotrophoblast and an outer syncytiotrophoblast. The embryoblast forms two layers, the epiblast and the hypoblast. These two layers together are known as the bilaminar embryonic disc. The syncytiotrophoblast is highly invasive. It burrows into the endometrium and starts to secrete the hormone HCG, human chorionic gonadotropin. This is the hormone detected in pregnancy tests, and its crucial role is to maintain the corpus luteum, ensuring continued progesterone production. By day 9, the blastocyst is fully implanted. Spaces called lacunae appear in the syncytiotrophoblast, and the amniotic cavity appears within the epiblast. By days 10 and 11, the syncytiotrophoblast erodes maternal blood vessels, establishing the early uteroplacental circulation. Around the same time, the extraembryonic mesoderm forms, filling the space around the amnion and primary yolk sac. On day 12, this mesoderm splits, creating a new space called the chorionic cavity. This brings us to week 3, and the pivotal process of gastrulation. The hallmark of gastrulation is the appearance of the primitive streak on day 15, at the caudal end of the epiblast. This structure establishes the body's craniocaudal axis. Cells of the epiblast then invaginate, or migrate inwards, through the primitive streak. This invagination happens in waves. The first wave of cells displaces the hypoblast to form the definitive endoderm. The second wave forms the intraembryonic mesoderm. The remaining cells of the epiblast now become the ectoderm. With this, the bilaminar disc has been transformed into a trilaminar germ disc, composed of ectoderm, mesoderm, and endoderm. These three germ layers will give rise to all tissues and organs in the body. Following gastrulation, the mesoderm organizes itself. A key structure to form is the notochord. It arises from epiblast cells migrating cranially from the primitive knot. The notochord is critically important because it serves as the primary inductor for the nervous system. Its main role is to signal the overlying ectoderm to thicken and form the neural plate, which is the precursor to the central nervous system. A remnant of the notochord persists in adults as the nucleus pulposus within our intervertebral discs. The process of forming the neural tube is called neurulation, which begins around days 16 to 18. The neural plate elevates its edges to form neural folds. These folds then move towards each other and fuse in the midline, creating the hollow neural tube. As the folds fuse, some cells at the crest of the fold separate to form the neural crest, a remarkable population of cells that will migrate throughout the body to form diverse structures like sensory ganglia, melanocytes, and parts of the skull. During this time, around day 20, the first somites appear. These are paired blocks of paraxial mesoderm that form on either side of the neural tube. They appear at a rate of about 3 pairs per day, eventually totaling 42 to 44 pairs by the end of week 5. Each somite differentiates into a sclerotome, which forms bone, a myotome, which forms muscle, and a dermatome, which forms the dermis of the skin. As neurulation progresses, the flat embryonic disc begins embryonic folding. Initiated around day 21 and completed by day 28, this process folds the embryo in two directions, longitudinally, creating a head and tail fold, and transversely, creating lateral body folds. This transforms the flat disc into a three-dimensional, C-shaped embryo, enclosing part of the yolk sac to form the primitive gut tube. The embryonic period, from week 3 to week 8, is a time of rapid organogenesis. On day 22, a milestone is reached, the heart begins to beat. This is the earliest functioning organ. The neural tube continues to close, starting in the cervical region and zippering up towards the head and down towards the tail. The ends, called neuropores, close between days 25 and 27. Failure of the neural tube to close properly can lead to defects like spina bifida or anencephaly. Now let's examine the placenta's structure. The maternal side of the placenta is formed from the decidua, which is the modified endometrium of the uterus during pregnancy. Specifically, the decidua basalis lies deep to the conceptus and forms the maternal part. The fetal component comes from the chorion, specifically the chorion frondosum, which is the bushy, villous part of the chorion. The placenta is therefore a fetal-maternal organ. Maternal blood fills the intervillous space, bathing the fetal villi, but the maternal and fetal bloodstreams never mix. From week 4 onwards, we see significant development in the head, neck, and thorax. In week 4, the pharyngeal arches appear, which are precursors to many structures in the head and neck. In week 5, the tongue begins to form, and by week 8, the ears and eyes are developing. Palate formation and fusion occur between weeks 7 and 12. In the thorax, the primitive heart tube, which formed in week 3, begins looping in week 4. Septation, the process of dividing the heart into 4 chambers, occurs between weeks 5 and 8. The lungs also begin to develop from the foregut in week 4, progressing through several stages of maturation that continue until well after birth. Finally, let's look at the abdomen and limbs. The gut tube differentiates into the foregut, midgut, and hindgut. A notable event occurs in week 6 when the rapidly growing midgut herniates into the umbilical cord. It returns to the abdomen in week 10, undergoing a complex rotation as it does so. The limbs begin as small buds in week 4. The upper limbs appear first, followed by the lower limbs a few days later. By week 8, the limbs have undergone significant rotation. The upper limb rotates 90 degrees laterally, so the elbows point backward. The lower limb rotates 90 degrees medially, so the knees point forward. This establishes the adult orientation of the limbs. Alright, that was a lot of information, so let's do a quick recap of the most critical points. Remember, primordial germ cells originate from the epiblast in week 2. Gastrulation, the formation of the 3 germ layers, is marked by the appearance of the primitive streak on day 15. The notochord is responsible for neural induction, leading to the formation of the neural plate. A major milestone is day 22, when the heart begins to beat. The neural tube closure starts on day 22 and is completed by day 27. The entire embryonic period, where all major organ systems are established, spans from week 3 to week 8. We've covered the core concepts of early embryology. Of course, this is a complex subject, and you may have questions. If you have any doubts or need clarification on any of the topics we've discussed, please share them in the comments section below. I will do my best to answer them. Furthermore, if there are specific topics within embryology or any other medical subject that you would like to see covered in a future video, please leave your suggestions. More explainer videos are on the way, and your feedback helps in creating content that is most useful for you. If you found this lecture helpful, please take a moment to like the video, share it with your classmates and colleagues, and subscribe to the channel for more high-yield medical content. The detailed PDF notes used for this session will be made available soon, so keep an eye out for that. Thank you for your time and attention. Stay curious, and I'll see you in the next video.